Hello, and welcome to today's webinar on the design and evaluation of complex sequential analysis trials. My name is Brian Fox. I'm a research statistician here at Endpoints, and I'll be your host for this webinar. So the agenda for today is to begin by discussing complex group sequential designs. Note that while this webinar will be talking a little bit about sequential designs, we will be focusing mainly on group sequential designs since they are the most common in practice. So I'll be touching on a few different kind of complex group sequential designs at that stage, touching on the advantages, some of the complexities that can occur within them. And then I will be moving on to our second section, which is group sequential design simulation, or I should say simulation for group sequential design characteristics. So during this stage, I will be discussing what simulation for group sequential design characteristics involves, kind of the advantages of it, when one might use it, and kind of the regulatory view on it, for example. And hopefully that will give you a bit of an overview on that. And then for the majority of that stage, I will be going through a practical example within Inquiry, just as a means of explaining each of the steps in detail. After that, I will be moving on to a group sequential design example. And also I will be trying to explain why group sequential designs for survival endpoints are very complex, especially when put into practical terms. And I'll explain how to go about conducting one of these trials. And once again, I'll be using Inquiry to kind of explain the steps involved within that. Then to conclude, I'll finish with a little discussion of all the things that I will have covered throughout the webinar, and provide some supplementary information for anyone who wants to go through it. I should mention that you should feel free to ask questions throughout this webinar through the chat function. I will not be leaving time at the end for questions. The hope is today to be done well within the hour, just to mention that. But yeah, if you have any technical issues or anything like that, do feel free to get in contact and we'll do our best to get those sorted for you. Okay, so I think that's all the housekeeping, hopefully. So just to give you a quick introduction to Inquiry, for those who aren't aware. So Inquiry is kind of seen as being the cost-effective and easy-to-use trial design platform, which can be an essential tool in optimizing your clinical trial. So Inquiry is known as the complete trial design platform to make clinical trials faster, less costly, and more successful. Inquiry is one of the most widely used sample size and power calculation softwares available, and has been on the market for well over 25 years. Okay, so just to give you a very brief overview of our licensing here in Inquiry, we basically have 4 different tiers, where each progressive tier contains all those below it. For example, the Pro tier here contains everything in Base and Plus, in addition to the additional tables or designs within Pro. And indeed, Pro is the one that contains our adaptive designs and tools for monitoring or adjusting trials. So that's the one that we'll be most focused on today for our examples. We do also have an Expert tier, which contains our prediction tool, which allows you to predict enrollment milestones for survival and non-survival endpoints. Okay. So now let's get into complex group sequential design. So I'll assume that most people here are quite familiar with group sequential design since you've joined this webinar. But just to give a brief overview and ensure that the terminology I'm going to be using is very clear and familiar to everyone, I'll just give a brief overview here. So sequential trials are basically just those which allow for accumulating data to be monitored and analyzed while the trial is still ongoing. So you can have fully sequential or group sequential designs. Fully sequential essentially just analyzes the data as it comes in continuously, and so after each subject's data accrues. Whereas group sequential trials analyze different cohorts or groups of subjects at certain interim analyses. So that's where the term group sequential comes from. So at each of these interim analyses, trial can either stop early or adjustments be made to the trial. The most common criteria for stopping would be stopping for efficacy and or futility. From a regulatory point of view, it's actually probably worth noting that the timing of these interim analyses and also any efficacy or futility boundaries should be pre-specified within the study protocol in advance. Okay, so the main benefit of these types of analysis or interim analysis, these types of designs, is that they provide a large degree of flexibility to the trial, and they facilitate interim decisions or modifications to be made to the trial while it is still ongoing. However, this flexibility does come at a cost, by which I mean we need to account for the effects of conducting multiple interim analyses, as we are essentially giving ourselves multiple chances to succeed, which invites the issue of multiplicity of testing. So naturally, the type 1 error rate would be inflated. So multiple methods exist for combating this issue. I'll assume once again that most of you are familiar with these. We do have multiple other webinars on this topic, actually, I should mention, hosted by both myself and many of my colleagues. So the topic of group sequential design is discussed a lot within our learning materials. Do feel free to just send us a message if you want to get any of those sent on to you directly, or just take a look at our website. So to get back to it, I suppose the main type of group sequential design that we'll be looking at today, and indeed the most common in practice by far, is the error spending approach, or the Land-Metz error spending approach, which it's often known as. And this allows one to spend your type 1 or type 2 error, where type 1 error relates to your efficacy or stopping for efficacy, and type 2 error relates to stopping for futility, and allows you to spend this across each of your interim looks using a spending function. So a spending function is essentially just a function which allocates portions of your total error to each of the specific interim analysis, in addition to your final analysis, obviously. So yeah, that's a basic overview of that method. Obviously, you have many others like Haybittle-Peto, Wang-Tsiatis, or Pamplona-Tsiatis. You have your classical designs like O'Brien, Fleming, and Pocock, which many of you might have heard of. Although I should mention that very often when one hears about O'Brien, Fleming, or Pocock designs, and generally, or well, a lot of the time, it's actually an error spending approach, and so that's being used. So there'll be some kind of analog to the O'Brien, Fleming, or Pocock design that's being used within the error spending approach framework. There are also more kind of flexible designs like unified family or Whitehead design, which actually could be argued to be part of the more sequential design or fully sequential design rather than just group sequential design. And sometimes that's referred to as triangular or Christmas tree designs, which you may have heard of. And then once again, multiple others, but we won't get into those right now. So complex group sequential design. So what I'm referring to when I say complex group sequential design is essentially a group sequential design which has an added layer of considerations to be made about the trial or design, which add to its complexity. It's very simple, nothing too complicated in the naming anyway, but different story when it comes to the actual designs. So the first one that I'll discuss here is survival group sequential designs. Might sound simple on the surface, where like survival or time to event endpoint is quite commonly used in practice, especially in oncology trials. But when it comes to group sequential designs, the added layers of complexity that a survival trial brings, or design I should say brings, have an even greater impact on the group sequential design. For example, like your patient follow-up, Will you be following each patient until the end of the study or just for a fixed period? So that has, as you can imagine, quite a large effect on the group sequential design, where the interim analyses have to be based on a level of information, so statistical information. So hopefully many of you will be familiar with that term within the context of group sequential design. If not, I'll be explaining it later when I go through the example. But yeah, so basically by that I'm referring to in a normal kind of means or proportions endpoint trial, and you'd be referring to your sample size as being the information, but within the context of a survival group sequential design, the statistical information will be referring to the events. So you'll be conducting your interim analysis when a certain number of events have been reached. So as you can imagine, if your patient follow-up is changing, or indeed if you have different accrual or survival or dropout patterns, which is actually my third point here, that will have a huge effect on when these interim analyses will occur, which makes things quite a bit more difficult to kind of predict, as you can imagine. Another point worth noting here is the idea of non-proportional hazards. So this adds a very large degree of complexity to certain survival trials, and group sequential survival trials are no different. So in the kind of non-sequential setting, you'll have multiple different arguments coming from multiple different views about how to deal with non-proportional hazards. Some just argue to use the Cox model or log rank model, or weighted, I should say, or stratified. Others will recommend using max combo method or Sequential or Apple method. But whatever you're doing, it does become quite a bit more complicated in the group sequential setting, and not as many methods that I'm aware of anyway exist for dealing with the issue of non-proportional hazards in the group sequential setting. But then, yeah, I suppose it is worth mentioning, like the stratified analysis, that is just a technique for dealing with the violation of the proportional hazards assumption, where you allow each group of subjects to have its own baseline hazard function, essentially. But yeah, definitely worth considering. And I will be discussing survival trials in a bit more detail later, so I won't spend any more time on that here. So the idea of an adaptive group sequential design, as you can imagine, adaptive, it does bring its, that word alone tends to bring a few added complexities. One of the main ones worth mentioning here is the type 1 error control. So that becomes quite a bit more of an issue in certain cases when you have adaptive group sequential designs. So by adaptive group sequential designs, I'm referring to a group sequential design in which certain adaptations are made to the design itself, not just the trial, in case that makes things a bit clearer. So yeah, the main one here would be the idea of having multiple different treatment arms. One of the most common methods in that area would be the multi-arm, multi-stage group sequential design, or the MAMS group sequential design, which is put forward by Gosh et al. In 2017 paper, I think. But it's very, very similar to the idea of drop the loser or pick the winner, which many of you may have heard of. And indeed, one of the more common examples of this type of trial would be the Stampede trial. And that'd be very similar to the graphic shown on the bottom right here, where you can see there are multiple different treatment arms, and a common control arm. And some treatment arms are being dropped at interim stages, and some are being added. So it's quite complex, as you can imagine. And so that would be one, actually, I would argue, one of the more complex types of group sequential designs from a practical view anyway. Maybe not conceptually. Also, the idea of sample size adaptations or re-estimations is definitely quite complex in the context of group sequential designs, where it could be either blinded sample size re-estimation or unblinded sample size re-estimation. Usually, we're referring to unblinded sample size re-estimation in the context of group sequential designs. So this also becomes even more of an issue in the case of survival endpoints. And once again, I'll discuss that a bit more later. Also, you can have different patient subgroups. That's obviously added a layer of complexity. And then often there are Bayesian approaches mixed in with the group sequential designs, and understandably so. It does help a lot with explaining the uncertainty that's inherent in a lot of these designs. So multiple different, very powerful and somewhat complex Bayesian designs do exist, and I believe are becoming more common in practice, although maybe not as much at the phase 3 or regulatory phase. Okay, so now let's move on to our second part of this presentation, which is to go into simulation for group sequential design characteristics. So essentially, it's very simple. All you're doing is using simulated study data to predict group sequential design characteristics. So this is obviously very useful, as you can imagine, especially in understanding more complex group sequential designs. So one of the main things it will be used for is verifying your original kind of design aspects, your closed form design. So by that I mean you could verify your type 1 error. So by simulating under the null hypothesis, you could verify your type 2 error or your power by simulating under the alternative hypothesis. You can also check your expected sample sizes, your stopping times. You can check when you expect to have a certain number of events if you're doing a survival analysis. And then also you can use simulation directly in calculating boundaries or other group sequential aspects, although it's generally not used for that as much, or quite rarely, I would say. And then also another benefit of simulation is that you can use it to explore alternative design or study scenarios. Essentially, you can evaluate lots of different alternative hypotheses. Like, for example, if the treatment effect is half of what you expect, then what effect would that have upon the trial? That's very easy to do within a simulated trial, where you don't have to worry about a lot of the constraints that closed form analysis might have. In addition, you can make changes to your interim analyses or your interim information fractions, and see the effect that would have upon the trial quite easily. And once again, this is particularly important in the area of survival analysis, which I'll discuss a bit more later. So I should also mention that regulatory views on simulation are usually quite positive. So I suppose I should mention that the FDA, for example, has multiple different guidance papers on computer modeling and simulations. And actually, I should mention here that while I'm primarily using just the word simulation, I am also kind of including computer modeling in a lot of the points I'm making here. And indeed, I should mention that I've included in the references some FDA and EMA workshops and guidance papers on simulation in case they're of use to anyone here. So the FDA themselves actually just view modeling and simulation as a very important part of the regulatory review process. So they're using it themselves in order to review different trials. And this view is shared by a lot of different other regulatory agencies. For example, the EMA has hosted multiple different workshops on modeling and simulation. And yeah, once again, I've included some of these in the references. But I suppose the main thing to emphasize here is that regulatory agencies tend to stress the use of a standardized modeling and simulation method, which can account for issues such as internal or external validity, and also control the bias or the type 1 error, especially in a confirmatory setting. Okay, so moving on. I'll just quickly touch on some of the considerations to be made if you're actually designing a simulation or simulated model for analyzing group sequential design characteristics. So I've split this into 4 different steps here in order to make it as easy to understand as possible. The first step that you would likely come across if you're designing a group sequential design simulation is what model assumptions you want to make. So just regard to your kind of design parameters and your allocation parameters. So I've included just a screenshot from Inquiry on the right here, where you can see you'd be specifying your means, your standard deviations, your sample sizes, all very simple. And then also you can specify your randomization routine. So that could either be being fixed by your sample size ratio, or it could be a complete randomization. So a complete randomization would be, in this case, where your sample size ratio is 1, would be the equivalent of randomly allocating each subject to the control or treatment group in the 2 mean case. Okay. And then the second step will be to specify your design inputs. So by this, I'm referring to things like your standard deviation definition. So would you want to have a pooled or unpooled analysis? Are you saying your standard deviation to be the same or not the same between both groups? Or are you going to use your test statistic as either a Z statistic or a T distributed, T statistic? And the kind of implications of the variance as a result. And you can obviously choose between 1 or 2 sets of tests, a simple number of looks, also once again simple, and then also to have your accuracy or futility bounds. So then at the third step, you would be specifying your group sequential boundaries and kind of the information fractions that you expect these boundaries to be used at. Once again, quite simple. I'll be showing that later, so hopefully that will become clearer then. And then at step 4, once again, nothing too complicated. You just be specifying your simulation controls. So these would be things like your the number of simulations you want to run, and also like the seed, the random seed that you want to use for your simulations, if at all. Very often they're just not specified. So then some of the questions that arrive from these steps would be, how do you justify your population assumptions? For example, like how do you justify choosing a pooled or unpooled analysis? Like, do you expect your standard deviation to be the same between both groups? In addition, you have to think about how to avoid your extrapolation issues. So once again, I mentioned in the previous slide, but you have to ensure that you're using standardized modeling and simulation methods to account for issues like internal or external validity. And then finally, you have to think about how to model your survival or accrual process, since that can obviously be quite difficult to predict. And obviously, I think I mentioned it earlier, Inquiry, we do have a product or a tool called Inquiry Predict, which aids in this process, as it can get quite complicated quite quickly. Once again, adding to the complexity of a complex group sequential design. So yeah, now let's finally get into our example. So this particular example is one that many of you might be familiar with if you tune into our webinars. It's one that I've chosen since we're not too worried about the actual example itself. We're more worried about how to simulate it. So I'm not going to actually spend any time going through this. Feel free to do, you'll be allowed to download the slides, and also I believe watch this presentation after it finishes. So you feel free to go back and take a look at this if you're interested. But it's nothing too complicated here. Okay, so, oh, apologies. So now let's go into Inquiry. So we're going to be using this here in order to go through this design. You can see that in the example, we have a sample size of 109 per arm. It was originally a 2-sided test, but you can see that here I've just switched it over to a 1-sided analysis. So this is just a replication of the original design. We see we get our sample size per group of 109. Then if you have the type an error and have a 1-sided test, you can see you get the exact same results. Then in the third column here, I've just added a futility spending function as compared to just efficacy spending function. I've just used the O'Brien-Fleming beta spending function or futility bound, since this would probably be the most common. And once again, this is the Landametz error spending approach rather than the actual O'Brien-Fleming kind of classical design. And also we're going to be assuming non-binding futility bounds, which is to say that you don't have to stop for futility necessarily. if your design crosses the futility bound at your interim look. So yeah, then just to make our simulated example a bit more interesting, I suppose, for lack of a better word, I've increased the number of looks up to 3. And you can see that that only requires an extra 5 subjects per arm. And everything else is the same here. So this is the one-sided analysis. You can see the boundaries here, your efficacy and futility boundaries, where if you're below these, you stop for futility. Above, in this blue region, you'd be stopping for efficacy. But just to illustrate it, because it's a kind of interesting point, I feel, if you had a two-sided analysis here, you can see that the group 1 and group 2 sample sizes isn't the exact same as the one-sided case. And that is because you can see here that your futility bounds cross, or well, maybe that's not as as evident as it might seem. But if you go to the plot here, the boundary plot, you can see that these are both set to 0. And essentially the reason for that is that within the software, or if you were to calculate these boundaries yourself, you would find that the futility bounds would have crossed. So your upper futility bound here would have crossed the x-axis of 0, or the 0 line here, and then your lower futility bound would have been above it. And so in Inquiry, we decide to set these to 0 since we feel that's the most practical approach. And essentially what you're doing there is just skipping the futility analysis at that look. Because if your futility analysis are both at 0, or your futility bounds are both at 0, then essentially that's like saying you can't be within the futility region. So yeah, that should be like if you go to the error spending plot here, for example, go to the first look, you can see that the red line is at 0 in this case, which is, corresponds to the right-hand y-axis here. So hopefully that kind of helps a little bit. But yeah, for our purposes, what we're going to look at is this example here. So if I go back to this tab, you can see there's a button, GST simulation. And essentially this button allows us to access the group sequential trial simulation tool within Inquiry, where if you use this button, it will inherit and bring across a lot of the values that are within your group sequential trial within this table, or design, I should say. So if I click this, you can see that our means and sample sizes, etc., are all brought across. And indeed, the information throughout the trial here, with the exception of some assumptions, will all be the same. And you see our futility bounds have all been brought across, our sample sizes and our information times or fractions. So just to mention, that's kind of how it works in Inquiry. But if you didn't have if you didn't have a group sequential trial that you were basing your simulation off, you can use our assistance menu here to get at the group sequential design simulation tool. And in that case, all you have to do is decide on the endpoint, the number of groups, and the analysis method that you're interested in. So you can see if I click this, it will bring us essentially into the exact same tool or steps, but it will be completely blank rather than having been pre-filled like it is here. So yeah, now let's actually do a simulation and show how this works. So I won't make any modifications for our first example here. You can see that all this is as you expect. I'm just going to be assuming the pooled known case. You can also choose to have unpooled known, so the set statistic, or once again, you can have the pooled or unpooled t statistic. Yeah, now let's keep going. I won't change the boundaries or anything like that here. We're just going to click run. Okay, and so you can see that our power is roughly similar to what you'd expect. Our power is 80, actually 80.2. This is lucky because I haven't specified the seed as you can imagine. This is just completely random. Since I haven't specified the seed, this could be anything, obviously, when it comes to simulation. But you can see that our power has turned out to be 80.5. So very, very close to our original power. So I suppose I should probably explain what this report is. So The report shown here is what we like to call our simulation report. This is essentially a big summary of the simulated data and the parameters that were used to generate that data. So you can see that we have our designer parameters, you have our allocation parameters, our distribution parameters, and our simulation summary. So simulation summary is essentially a summary of all the different simulation control parameter values. And also you can see that even though I didn't specify the seed at this step here, it is outputted. So if you do want to go back and replicate your results, like if I were, for example, if I was to highlight this and throw it in here, we should get the exact same results. So you can see we once again get the exact same power and all these are the same. So then on the right here, we have our boundaries table, and it's essentially a summary of our futility and efficacy boundaries, if applicable, our information fraction, and then analysis at which they apply. We also then have our number stopping table. So this is kind of like a summary of the simulation itself. This kind of shows how many of the simulated trials stopped for efficacy or futility at each interim look and the final analysis. So for example, here you can see that the number of trials that stopped for efficacy at the first interim look were 2.44. The number that stopped for futility at the third, at the final analysis were 819. Because we had 10,000 simulations, you can see it's a nice even number, where that's 8.19% of the cases. And you can see that our power is just our efficacy percentage, which is to say that under the alternative hypothesis, you expect this number of trials to stop for efficacy. And so that's our power. Very simple, nothing too complex. And that kind of illustrates actually just how easy it is to explain one of these designs to a non-statistician or any other stakeholder in a trial, which is one of the main benefits of it. Okay, so actually I should probably go back to our example here, and I'll just explain some of the modifications or hypotheses that I'm going to test here. So let's just test the case where we want to verify our type 1 error, for example. So obviously to do this, we're going to be assuming the null hypothesis to be true, which is to say that the mean difference is 0. So if I go in here, we go back to, actually I'll create a new one, it's pretty easiest. So if I go in here, and now we're just going to set our null hypothesis to be, our mean difference to be 0. So I won't change anything else. The rest of this will all be the exact same. And we should see, once again, I'm not setting the seed, just so you know this is all honest example. Yeah, you can see that our average power is 2.22. So we would have expected 2.5 to be the power, since this should be the exact same, or sorry, this should be similar to the type 1 error rate from our original design, which is 0.25. So yeah, like if you were to set the number of simulations to be quite a lot higher, then you should find that it would narrow in on a 0.25 or 0.25, I should say. I'm not going to do that here because it takes a little while to run, but yeah, one may choose to do that if you wish. Okay, so now let's check what some changes to the treatment effect can have, since this is very, very easy to do within a simulation. So we're going to go back to our inputs, and I suppose we've technically done this a little bit already, but instead of I believe it was 2.3 before, let's just imagine that there is a very narrow kind of mean difference. So let's say minus 1.8. So very, very small, still the same direction, obviously, but quite a bit smaller. So what we expect here is that when we conduct the simulation, we'll see a lot more trials stopping for futility and not as many stopping for efficacy. So if I proceed through, once again, not changing anything else, we can see already actually in the update that the power has dropped significantly. And we can see a huge increase in the number of trials stopping for futility compared to before, where we had 1,948 stopping for futility. Here we have 82%, roughly 82.39. So yeah, you can see that was very, very quick to do, very easy. It's not just a, a sign that the software is easy to use, it's a sign that simulation is very easy to use, very easily understood as well. So, okay, I won't bother checking the larger mean difference case. It's very easy for you to understand. Let's check some changes to the variance. So if you were to go back to our first example once again, and instead of, actually I'll redo it just to make it a bit easier. So I keep clicking into that one. That's our survival example. We'll be getting into that later. I keep teasing it. But yeah, so here let's just assume that our group 1 standard deviation ended up being, let's say, a little bit larger than expected. So let's say it was 2.8. Then what we should see is that this results in a reduction in power. So everything else will be the same. You can see that our power is reduced actually quite a bit, even more than I thought it would be, to be honest, from 80 down to 66 roughly. And we can see that once again, our futility percentage has increased quite a bit. So that's to be expected. Once again, this is nothing too complicated, but it does show how easy it is to analyze this within simulation. So now let's take a quick look at changes to the information fractions. So this is especially important if you had a survival trial. I'm teasing what's coming on later, but if you did have let's say an early bias, which is to say that the majority of the sample size happened early on in the trial, or sorry, at the early analysis. So if I go back, actually, once again, I'll have lots of tabs open, but I'll just keep creating them for the sake of clarity. So if we're to proceed through and at this look here, if we were to change our analysis time to be a lot earlier on, so let's say it's 0.1 and 0.4, you can see that our boundaries will be becoming a lot earlier. A lot more of the trials will be stopping. Or sorry, probably less than the trials will be stopping early on, since we wouldn't have enough evidence in that stage. And so you can see that, yeah, once again, our power is decreased by quite a bit. So yeah, it's very easy to kind of conceptualize this. You can see the kind of effect quite clearly there of having earlier interim analysis. But if you were to have later interim analysis, so let's say 0.8, or well, let's say 0.7, and then 0.85. Oh, apologies, I have to go back. Or actually, I'll just create a new one. Can never have too many tabs. But yeah, so if you were to have, let's say 0.8, or sorry, 0.7. Oh, once again, this is going to happen again. Once again, never have too many tabs. It's more the better. So if we go here and change this to, let's say, 0.85 and this to 0.7, we should see that now, since it's far higher sample size at these later stages, the proportion which we're able to stop for efficacy should increase a little bit. And indeed, we do see that it's 83.6, so a marginal increase, nothing too extreme. So yeah, quite easy to explain, understand. A stakeholder can very much understand how these numbers are resulting from the changes made. Okay, and then also actually I should mention that you can see your average sample size here. You could calculate this analytically, obviously, but it's very easy to do it within a simulated trial. So, for example, if we can go back to our first simulation example, you can see that our average sample size was quite a bit lower. It's 190. But then if we proceed to, actually, I don't know if it's included here, but yeah, so the case where we had a far lower power, you can see that our sample size is once again quite a bit lower because we're stopping for efficacy in a lot more cases early on in the trial. So yeah, basically just worth mentioning, that's included here. It's quite useful information. Okay, and then the final thing I'm going to go through here, since I am aware I'm spending quite a bit of time on this, is the notion of having complete randomization. So instead of having the randomization being fixed by the sample size ratio, let's think about what would happen if it was complete. So in this case, we can see that once again, we wouldn't expect the power to change very much here. Essentially, if you're running a huge amount of simulations, you'd expect both to be the exact same. But what we can see is if we go into our summary data here, which is a summary of the kind of per look data from within each simulated trial. So this is kind of each of these simulation IDs here. They refer to a simulated trial. And then our per simulation subject data gives you the the data from that simulated trial. So if you can obviously save these data sets if you wish from within Inquiry. But if you're to take all the responses here for the treatment ID equal to 1 case, you'll get back these results here. So you can see that our group 1 and group 2 sample sizes are very different here. So, at look 1, we have 34 versus 46, despite the fact that we had our sample size ratio set to 1. The reason for that is that we're doing a complete randomization. Whereas if I go back to our earlier examples, you should see that these will be almost the same, or the exact same, so 40 and 40. And that's because the sample size or the allocation is fixed by the ratio. Okay, so I suppose I should mention actually just while we're here, the other information contained within this data set. So it will also tell you what happened at each interim look. So you can see here that if you don't stop for efficacy or futility, then you can continue, or the trial has continued here in this particular simulation. And then at the second look, it has stopped for efficacy. Whereas in the next simulated trial, number 3, it has stopped for futility at the first look. And the reason for that is that our test statistic of 0.343 is within our futility bounds. So, above our futility bound, since this is one-sided and it's a negative mean difference case. If the mean difference was positive, obviously these boundaries would be flipped, or well, multiplied by -1, let's say. Okay, and then like if there's any specific questions in this, do feel free to ask. I know I'm going through this quite quickly, but essentially there's nothing too complicated here. As always in Inquiry, we have help cards on the right, which will explain anything as best we can. But feel free to get in contact if anything isn't clear. If you'd like me to go into more detail on anything, I can send on more information afterwards. And then also, actually, I should mention that within Inquiry, we do also provide a rejection plot. So this is essentially providing a graphical view of the number selected for efficacy, for futility, or continuing at each interim analysis and the final look. You can see that obviously at the final look, we have to stop for efficacy or futility since our boundaries are the same. You can see 1.993 for both. And that is why we have to make a decision at the final analysis, obviously, and why there's no continue section there. Okay. And then also we do have a boundary plot. This is kind of similar to our group sequential design boundary plot, where you'll get a synopsis of the actual boundaries in the table below, and multiple different options can be made with regards to editing that plot. But I won't go into any of those here. Okay, so I'm aware that took quite a while, but hopefully explained simulation pretty well, or well, hopefully anyway, and gave you an idea of how you can do that, not just within Inquiry, but even within whatever software you're using. You can see it's quite easy on the surface, maybe not so easy to code or to actually put into practice in software, but very easy to understand, very easy to kind of conceptualize how it would be done. Okay. So now let's move on to our third part of this presentation, which is survival for group sequential designs. So here I've just taken a slide actually from our previous webinar from last month, so anyone who saw that will be well used to any of these formulae and what's kind of discussed here. But just to quickly discuss it, there's quite a lot to unpack when it comes to survival analysis. So the power for a timed event endpoint design is related to many different choices. So you can obviously have a parallel design, one arm, etc. You could use the log rank test or the Cox proportional hazards model, or weighted statistical or stratified versions, etc. You can change your hazard or your endpoint. It could be the hazard ratio. It could be your progression-free survival, your overall survival, etc. You can change your survival distribution from the most common to, I suppose, be the exponential. Well, exponential would be the most common, and then viable would probably be the second most. And then also, yeah, it's definitely worth noting that the power is not driven by the sample size in the survival case or time to event case. It's a mistake that's sometimes made. It's driven by the events, and that's clearly shown in the formulae on the right here. And you can see that it's, well, I suppose one of the most common ones would probably be the Leakine and Fuchs formula. But yeah, essentially, it's very important to keep in mind that the power is being driven by the events, not the sample size. And that kind of ties into my point from earlier, that in a group sequential setting, it's your events that are linked to the information and not the sample size. So yeah, so the sample size determination is just basically interested in getting the number of subjects to reach that number of events. So some people call it like a meta model, which accounts for the accrual and dropout and crossover, etc. But yeah, like it's very easy to say these things, but actually putting them in practice, and like if you're actually conducting a group sequential trial where your first interim analysis, let's say, is after I don't know, 20 events, then you expect 50 subjects for those 20 events, and it ends up taking 100. It can be quite a difficult problem to overcome. So yeah, just to kind of touch on these problems and talk about some of the considerations that have to be made when you're conducting a survival group sequential design. I have this slide here. So I suppose one of the most important points is to note that due to the kind of unknown follow-up time, there's a lot more uncertainty about the interim analysis, which I've just touched on. But it becomes just very difficult to predict the time when interim analysis can occur, or well, can be very difficult. So it becomes common in many cases that more subjects will be in the, more events might occur in the active cohort when the interim analysis actually occurs, which can obviously affect your original, can be at odds with your original design assumptions. Another consideration to be made is whether to use progression-free survival or ordinary survival at each look. Like I should mention, you're able to switch between these, or well, maybe conduct progression-free survival at earlier interim analysis, which is shown on the right down here in this example from the European Patients Academy on a therapeutic intervention. So you can see that obviously here at the first interim look, they used progression-free survival as their endpoint, and then at the second it was overall survival, which would be preferable usually, overall survival, obviously. Okay. Oh yes, and then another thing to keep in mind is if you're planning to conduct unblinded sample size re-estimation, then this can have quite an impact on your kind of assumptions. So if you are planning to do one of these, beyond just the usual kind of practical considerations that are associated with unblinded sample size re-estimation itself, for the time to event endpoint, there are usually additional kind of trial integrity considerations to be made. For example, the Friedling and Korn paper here from 2017, and they discuss how access to interim information on the shape of the survival curves can jeopardize the perceived integrity of a trial if you're using sample size adjustments. So essentially, they demonstrated that when the magnitude of the true treatment effect changed over time, The interim information on the shape of the survival curves can be used to enrich the final analysis with events from the time period with the strongest treatment effect. So this obviously gives one the ability to kind of make the end of trial treatment effect larger, on average, based on the decisions about using the interim kind of data. So yeah, that is obviously pretty important to just be aware of. So another thing to kind of keep in mind is that survival group sequential designs often require additional assumptions. So, do you assume a constant treatment effect, or, is there non-proportional hazards? Obviously, this assumption is often made in survival trials anyway, but it becomes even more important in group sequential setting since it can be very specific to each of your analysis and, have a huge bearing on how likely you are to stop for efficacy or futility, especially maybe earlier on. If there's a delay in the treatment effect, for example, then obviously that would increase your chances of stopping for futility. So the final point I'll mention is just that if you're that your interim events are dependent on two factors. They're dependent both on the sample size and the time. So the time of that interim event or analysis, I should say. So if you increase the time, then this would bias later trends in hazard, whereas if you increase the sample size, then it would bias earlier trends. So it's definitely something worth keeping in mind, very important to be aware of. Okay, so now let's finally get into our group sequential survival example. So before, or well, I suppose I'll talk about it a bit here. Essentially, kind of similar to our earlier example, I'll be using an example from previous webinars. In fact, this is one used in last month's webinar for everolimus for advanced pancreatic neuroendocrine tumors. So I'll just be extending this to the group sequential and a piecewise survival case. So we'll be assuming that 5 looks are planned, so 4 interim looks and 1 final look. We'll be assuming efficacy and futility bounds are both present and they're non-binding, and we'll be using the Landau-Metz approach. We'll be assuming an exponential alpha spending function, just something a bit different to kind of show how it works. And we'll be using a parameter of 0.8, and that essentially kind of replicates the O'Brien-Fleming Landau-Metz approach, as we'll see, although not exactly. It's pretty similar. And then just to do something very different for the beta spending case, we're going to be very kind of non-conservative and use t-distributed boundaries. And we'll see, I'll show this later, show how that appears. And then we'll be assuming our survival time periods there's 3 survival time periods: 0 to 6, 6 to 18.5, and 28.25 months. And then we will be, this isn't exactly what we'll do from the get-go, but effectively we'll just have different exponential hazard rates and show how they change over time. We will be assuming a constant hazard ratio, I should mention, but I'll show that one in the example in just a moment. And we are going to assume some dropout just to show the effect that can have on a trial. Okay, so now, I suppose I have all these open, so I might as well show them to you. These are just replications of that original example, where 282 events and 176 and 92.6% power, just very similar to here. You can see the 92.6 power. It's just effectively justifying the original example, so nothing too important there. Here, this is a table we have which uses simulation. I'm barely showing it because we've talked about simulation quite a bit. My colleague Paul actually discusses this in a lot more detail in last month's webinar, so do feel free to have a look if you want to have a bit more information about that. But essentially, in this case, you don't have to have your constant hazards. You can change it if you wish, although doing this without adjusting for other things might be quite devil may care, one might say. So then finally, this table here is SD37. It's a new table in our recent release. And this, which actually only came out yesterday, I believe, depending on when you're watching this, But this table here effectively allows you to choose between having event-driven, which is to say kind of maximum follow-up, or a fixed follow-up. So you can choose to just have a follow-up for a fixed period. So in this case here, you can see that the original example here is giving a very similar power to before, 92.6, 92.7, very, very close. And then the next example had fixed follow-up of 5, which obviously reduces power since you're only following each subject for a maximum of 5 months in this case. But yeah, I'm just basically showing this to show kind of where I'll be getting my values from. So this table that I kept clicking into earlier, I kept teasing you with, this is GST3. I'll show how this works by doing it from scratch. So this is essentially just a group sequential design for survival, or 2 survival curves. So if I go back here, hopefully I can do this from memory. So it's 0.25. We have 5 looks, 3 periods, since we'll be having a 6-month period at the start as well. Leave that as event driven for the moment. Our hazard ratio is 1.5. And we will be using a spending function futility spending function. And I'll leave this for the moment while I enter in our group sequential spending function parameters. So we're looking for exponential alpha spending function, perhaps not as common as the O'Brien, Fleming, or Pocock, but quite flexible, actually very flexible. I'm a fan of it myself, but obviously you'd have to justify any choice of a spending function here. And then our second spending function, something quite a bit more different, is the t-distribution. So our efficacy parameter is going to be 0.8. You can see in the suggestions here, a value of 0.8 is expected to give similar results to the O'Brien-Fleming spending function. So our t-distribution boundaries are going to be, parameters are going to be 0.5, 0.2, and 5, I believe. And then our piecewise information. So we're going to be having interim analysis, we're going to be having piecewise time periods of 6, 18.5, and 28.25. And we'll be assuming that, just for the sake of example, we'll assume that there's very slow accrual for the first 6 months, and that the majority of it is done between the rest of it is done between 6 and 18.5 months, which means that there's 0 in the final period. To get this value here, I'll just go and copy this value from our previous example. That's just the exponential hazard rate based on our medium survival. Okay, so then we'll assume a dropout rate of 0. Actually, for the moment, we'll just assume no dropout, and then I'll show the effect that can have afterwards. Okay, so if we go back here, enter in our sample size ratio, and I suppose for the moment we'll be in keeping with the design, and we'll enter in our sample size as being 176. Okay, and so this calculates, and you can see our power is quite a bit reduced to before. That's just due to the fact we're conducting a group sequential analysis with 5 interim analyses. And we have quite kind of loose futility bounds here, where they're quite, well, relatively loose, one might say. So you can see that our power has dropped down from, 92.7 down to 82.3. So yeah, so that's the cost of doing a group sequential analysis. If I go into our group sequential report here, however, you should see that our expected sample size, scroll past it. Our expected sample size under both H1 and H0 is the maximum sample size. So we see that we're not actually stopping for FCM futility that often here. So we are very likely to get to the final analysis. That's what this is saying here. So I know there's quite a bit of information in this table. Actually, I'll first conduct an example with dropout, and then I'll go through it. So if we have dropout, obviously we expect our power to decrease, and also our total events to decrease, our sample size will obviously be staying the same since we're specifying those. So if I specify this, we come in here, once again, we'll specify our parameters. So 0.8, 0.5, 0.2, and 5, I believe. Was that correct? Yes. So yeah, and then for our piecewise information, we just copy and paste over here. And we can see, oh, and apologies, I meant to put in the dropout here. So we're going to be assuming quite a large amount of dropout. So 0.1, 0, 0, 0, 0.05, and 0.1. So we're going to be assuming that everolimus had kind of later trend in dropout, and that the placebo dropout rate was quite a bit earlier on. Okay, so now if we go back and enter in our ratio, the calculation will begin, and you can see that we have quite a large reduction in both the power and the number of events, as you'd expect, can have quite a large impact. So now I'll just quickly go through the group sequential report for this case. And I suppose most of this might be familiar from earlier group sequential webinars, so I won't really go through it. The main thing to note here is our trial information tables. So here you can see not just the information fractions and the number of subjects available at each of these analyses. You can also see the time that we expect it to take to get to a certain number of events. So you know, we expect it to take 13.01 or 0 months to get to 43 events, and then you can see it will take quite a long time to get to the total number of events, actually beyond the time allotted to the trial. So we never expect to get to that number of events within the time allocated to this trial, since our maximum time is 28.25 in this case, I believe. So we expect to actually only get to somewhere between 174 and 217 events. So then you can see also the dropout, so the amount of subjects we expect to drop out. You can see the expected amount of people left in the trial at any given time, which are available to have an event. And so, yeah, multiple different bits of information that are extremely useful for not just conducting the trial, but for kind of analyzing how the trial is expected to occur. Okay, so I just realized I'm quite close to being out of time, so I'll quickly finish up. So basically, this is just a quick synopsis of all the points I made here today. Basically saying there's multiple different complex sequential trials out there. Many have their own merits and kind of considerations to be made if you're conducting them. So once again, simulation, very easy to understand, maybe not so easy to kind of code, but very easy to implement once you have it kind of set up, and very easy or very useful for not just a verification of your original design, particularly group sequential designs or complex group sequential designs, but also in exploring different scenarios. So it's a very quick, very easy to explain tool in the statistician handbook. And then finally, we had a quick look at survival group sequential designs, which obviously have their own considerations, where in particular it becomes very difficult to predict interim timings, so interim analysis timings. You can also change the endpoint at different looks, you know, progression-free survival, change to overall survival. You can have different effects, varying hazard ratios at each look, although we obviously didn't explore that here. But yeah, once again, multiple different things to be aware of. It can become quite complex very quickly, but hopefully I've given you some tools, at least today, to kind of combat that complexity. I've only included this here in case anyone's interested in kind of learning more about what's included in the group sequential kind of updates that have been made to Inquiry in a recent year or so. So this is just a list of some of the things we've added. As I know, there's quite a bit there. I just encourage anyone to have a look if they want to, or feel free to get in contact with us at info@statcells.com, or you can get more info also on the website at statcells.com. And then also, actually, just to mention, if you want to get a demo, you can go to this link here, /inquiry or -demo, and it's free of charge, easy to do if you wish. Okay, so yeah, thanks for sticking with me up to this point. Hope you have a nice day, and thanks again.